We are preparing for the SeekOne DD single-cell 3' transcriptome experiment. Now we move on to droplet generation, where individual cells are encapsulated and barcoded. Before starting, place all reaction reagents on ice. Keep the barcoded beads and carrier oil at room temperature. First, take the barcoded beads out and let them equilibrate to room temperature for at least 30 minutes. Thaw the TSO on ice. Vortex. Spin briefly and keep it on ice. Thaw 3 times RT buffer and reducing buffer on ice. Vortex. Spin briefly and keep on ice. Keep the RT enzyme at -20 degrees and only take it out right before use. Place the instrument on a stable bench, power it on, and run the self-check. Now, prepare the reaction mix on ice. Add the reagents one by one. First, the RT buffer, then RT enzyme, followed by TSO, and finally, the reducing buffer. Pipette about 15 times to mix. Spin briefly. And keep the mix on ice. Before use, check the 3 times RT buffer. It should appear pink and clear. If the color changes or precipitation is visible, discard the reagent. Before loading, first consult the loading table. The highlighted zone indicates the recommended concentration range. The red values indicate the required cell suspension volume. The total volume is 80 microliters. This includes the cell suspension, nuclease-free water, and 35.4 microliters of master mix. To calculate the cell suspension volume, divide the targeted recovery number by 50%, then divide by the cell concentration. Next, subtract the cell volume from 44.6 microliters to determine the nuclease-free water volume. The recommended capture range is 500 to 12,000 cells per channel. Let's walk through a quick example. If the cell concentration is 1,000 cells per microliter and the target recovery is 12,000 cells, the required cell suspension volume is 24 microliters. Then subtract 24 from 44.6 microliters to calculate the nuclease-free water volume, which equals 20.6 microliters. Now the sample is ready for loading. Here are a few important notes for buffer preparation. Nuclease-free water may be replaced with the appropriate resuspension buffer. For nuclei samples, always include RNase inhibitor to prevent RNA degradation. For whole cells, RNase inhibitor is optional, but recommended for better RNA stability.